So the only thing I wanted to go over today was just the lab for today, which is the muscle physiology lab. And then if there's any questions about the exam for tomorrow, and that covers lectures 5 through 8. My goal is to get all the lab reports graded, the cardiovascular lab reports graded today. So you guys will have feedback on that before you start your next one. So make sure you read that feedback so that you are set up for success so that you can do better on that second lab report. And that one's due on Thursday. What else? The XFIZ topics, I would like to have those by tomorrow sometime. So again, if you guys want to start like a group, and maybe you already have, just like a group email to figure out who wants to be in which groups and what topics you're interested in, then we can kind of go from there and just email those to me, each group. Okay, so the lab, let me find it. It's pretty self-explanatory, but I'll kind of go through it. So for station 1, again, everyone should do this, and then you'll have your own data for this. Again, you can take a picture and include that with your handout or whatever you're handing in. But you're basically just going to write your name with normal with your hand after your hand's been submerged in ice water for 3 minutes, and then with your hand after it's run under hot water or something like that. To look at the difference in muscle contraction and dexterity and things like that. Station 2, again, everybody will do this, so it's just labeling the different components associated with the muscle, and then just make sure to pay attention to, there are some sneaky little questions hidden in here, so make sure you don't skip those. They're not like labeled well, I guess. But make sure you do answer those because I will be looking for those in the handout when you hand this in. Describe the steps for muscle contraction. Again, you got your picture here. So what I would do is do like, okay, number 1, this is what happens. Number 2, here's what's going on with, step number 2, etc. So you can kind of match up the picture with your description of how the muscle contraction occurs. Again, don't skip these little questions that are kind of snuck in here. The muscle fatigue section, again, everyone will do this. You should have all received a clothespin in your lab package. And again, you might need somebody to help you, or you could set up like a automatic timer if you can. I don't know how to do that. I'm sure it's like not that hard though. To time yourself for each of these. So you only get 5 seconds rest between each one. So you're going to do 15 seconds with your clothespin. Again, keep it on the same hand for the whole experiment, and then just write down how many contractions you get for that 15 seconds. The idea being that you should get fatigued and the number should go down. The strength decrement index, this is always confusing for people. So you're going to look at trial 1 in comparison to each other trial. So ideally, trial 1 should have the most contractions. So ideally, your graph should start, can you see my pointer?
Yeah.
Okay, so your graph should start kind of up here and then go down. Okay, and that basically means you lost strength. So again, that's kind of the idea with that. So you start with 100% strength, and then it tells us what percentage you're at in comparison. Histology, again, just kind of do your best to draw what each of those types of muscles look like. Again, I'm not like grading you on your artistic abilities or anything like that, but just do your best to kind of include all of those kind of components that we know about. And then the hand grip section, again, only if you've received a hand dynamometer. So I think I sent 3 or 4 of those out. We'll do this section, and then the results will be posted on the Google Doc for that. So please try to do that today if you can. Again, that should only take a minute or 2 to do this experiment, so that way people have all that information. If they wanted to start their lab reports today or tomorrow, they'll be able to do so. So again, with that, and I think I showed that in the video for using your equipment, but basically flexed position, your wrist is going to be turned in. Neutral position is just like a flat position, and then extended position, your wrist is kind of flicked back for each of those. So in reality, ideally, we want neutral to have your largest number, and then both the extended and flex position will be a little bit lower for those. Please record those in kilograms. I'm not sure if I included that on the on that Google Doc, but record those in kilograms if we need. Okay. And then the last little section, again, you might need somebody to help you with this, or you can have test this on somebody else, but again, you'll need all your own data for this one. You'll look at the circumference. So again, you should have received a tape measure with your package, and you'll do the circumference of the upper arm. So again, just try to get the widest portion of each of those sections of the body. So you'll do relaxed, and again, relaxed should be the same for the anterior and posterior, and then contracted. So like for upper arm anterior, how would I contract this? Like do something where I'm contracting the biceps, okay? So you'll measure the circumference there, and then look at the difference between the two, and then know the muscles listed, what muscles are activated when I contract the anterior portion of my upper arm. One other little hint, for the most part, all of these, your relaxed circumference should be less than your contracted circumference. However, with the calf sometimes, and even for the upper leg sometimes, we'll see that the relaxed is actually larger. I want to make sure I say that right. The relaxed is sometimes larger than the contracted version, which is confusing for people. For your arm stuff, the contracted version should pretty much always be larger than the relaxed version, but like for the calf sometimes, with the compression, it is actually smaller when contracted than when relaxed. So that's one thing to think about. Again, the reason for that is probably blood flow restriction when I contract some of those lower limbs. And then a couple more questions there at the bottom to answer. And then, so you'll hand that in, and then you'll also have a lab report to go along with this. The lab report will focus primarily on two things. So we'll look at the strength decrement index with the clothespin, and then also the wrist position with the grip strength. So the hand dynamometers, and again, 3 or 4 people are doing that particular experiment, okay? So statistical analysis for grip strength, we want to use repeated measures ANOVA for that. So there's 3 time points, and we'll have one group going through each of those time points. And then for the clothespin experiment for the strength decrement thing, you'll just look at your own data. Was it statistically significant? So again, for that, probably regression would be the best thing for that one. Again, same format as what you did with cardiovascular. So I would probably wait till you get some feedback on that lab report today before you start on this one. But the main thing that you really want to focus on in this lab report is the length tension relationship as it relates to those different wrist positions, okay? So again, when we think about actin and myosin overlap, which you'll learn about in today's lecture, again, think about how much actin and myosin are overlapping based on the position of the muscle, and that's really what you want to focus on here. And then lastly, again, you'll choose some disease that you want to, and find out what physiologically is going on with it, and then summarize 2 studies. Again, when we talk about summarizing those studies, think about what was the purpose of the study, how did they set up the study, what was the subject population used, what did they actually measure, and then go to, what were their kind of concluding remarks or concluding findings, that sort of thing. So you want 2 of those. Again, a nice big paragraph for each study is usually pretty good to kind of hit all your bases for that. Questions?
Not yet.
Okay. So I think that should be good to get you guys started on everything you need to for today. Exam tomorrow. Again, if you guys do have questions as you're studying and preparing for that, just text me or email me or whatever. Yeah, same format as what we've done before. And yeah, we don't really have a whole lot of need for these Zoom sessions anymore because I set it up as they're coming, the babies. But we'll just continue to meet as long as we can and just kind of, be able to touch base and review on anything that we need to. So yeah, so the main thing for today is just make sure we get going on those XPhys group projects. The environmental projects, I can't remember who our 2 subjects are, but I'd like to get that data done by Wednesday because you guys have presentations on those for Friday. And the groups are posted online for that.
Okay.
That's it. That's all I got.
Awesome.
All right. I'm going to get to grading. You get to studying. You're at work, so you're going to get back to
I am
work.
at work right now.
And yeah, we'll see you hopefully on Wednesday.
Yeah, sounds good. Thank you.
Yep. See ya.
Bye.